This is lecture 18 on gas transport and regulation of ventilation. We'll start our discussion on gas transport looking at oxygen transport in the blood. Oxygen is transported in two different ways in the blood. First, 2%, so very little, is dissolved directly into the plasma. That means 98% of it is bound to hemoglobin, specifically the iron ions on hemoglobin. Each hemoglobin can bind up to 4 oxygen molecules, 1 per iron-containing heme unit. And the hemoglobin binds when the pressure of oxygen is very high in the lungs, but releases oxygen when the pressure of oxygen is very low in the tissues. So once again, diffusion and the actual pressure of the gas is going to determine the direction of diffusion and allow for exchange. Now, when the hemoglobin molecule binds to oxygen, it is called oxyhemoglobin. This is a reversible binding that allows itself to detach or reattach based upon the rate of diffusion. Carbon monoxide, or CO, can also bind to heme units, making them unavailable for O2 transport. This is why carbon monoxide detectors are so important in households, as it truly is the silent killer, because carbon monoxide will bind to the actual heme units and end up suffocating you. Here we can see an actual structure of the hemoglobin molecule. We have the protein subunits and the iron ion within the heme unit. Remember that each hemoglobin can bind up to 4 oxygen molecules to each one of the heme units. When discussing oxygen, we need to learn a little bit more about the oxygen hemoglobin saturation curve. This curve is a graph that shows the hemoglobin saturation at different partial pressures of oxygen. Hemoglobin saturation is the percentage of heme units bound to oxygen at a given moment. So how much oxygen is bound to the blood? The hemoglobin saturation curve is actually going to be S-shaped. Now, this is due to the increased affinity that hemoglobin has for oxygen with each oxygen molecule bound. This is going to increase steeply until it reaches a plateau near full saturation. Hemoglobin is more than 90% saturated with a pressure of oxygen above 60 millimeters of mercury, so we can survive in a very wide range. This is the oxyhemoglobin saturation curve. You can see the difference at different points where the blood is in the body. The pressure of oxygen in active muscle tissue, the pressure of oxygen leaving peripheral tissue, and finally at the very top, the pressure of oxygen in the blood entering systemic circulation. Now note there is a very steep slope. This is where a very small change in the blood pressure of oxygen will result in a large change in the amount of oxygen bound. Hemoglobin responds most dramatically at lower pressures of oxygen. Small changes in the pressure of oxygen can mean large changes in hemoglobin saturation. Continuing from the actual diagram, there were some different points that were discussed. The pressure of oxygen, 95 millimeters of mercury, is the blood going to the peripheral tissue, is the blood going to the tissues. Here, hemoglobin is 97% saturated. Whereas leaving the tissues, it's going to be at a pressure of oxygen of 40 millimeters of mercury and is only 75% saturated. Here, substantial oxygen reserves are found in the venous blood based upon usage. At the pressure of oxygen of 20 millimeters of mercury, like in active muscle tissue, hemoglobin drops to only about 20% saturated. This indicates a release of a large amount of oxygen to the tissue and should make sense. Active muscle tissue is going to use the most amount of oxygen. Now, there are a number of factors affecting the oxygen hemoglobin saturation curve. Remember that another way to look at the oxygen hemoglobin saturation curve and what it means is quite simply of how readily the body can actually bind and use oxygen. So that means if it is a higher overall saturation, the body is better at actually binding oxygen. Whereas if it's lower, it's not as good or is having trouble doing that. Now, there are 3 factors that affect the oxygen hemoglobin saturation curve. They are blood pH, temperature, and the pressure of CO2. The blood pH is known as the Bohr effect. A pH decrease, or making the blood more acidic, is going to cause a saturation curve shifting to the right, meaning if we make the blood more acidic, the blood is less able to bind to oxygen. We know this happens, of course, during exercise. Our muscle becomes a little bit more acidic and therefore is going to have a harder time binding with oxygen. Conversely, if we have a pH increase or the blood is more alkaline, we have a saturation curve shift to the left. When the shift is to the left, that means we are more readily able to bind to oxygen and we are more saturated with it. Temperature. Higher temperature leads to hemoglobin releasing oxygen more readily. So it's not going to be able to bind as effectively and we release oxygen more rapidly. Again, like during exercise. So an increased muscle temperature is going to mean a lower oxygen affinity. So it's not going to be able to bind as effectively. Finally, a pressure of CO2. If we have an increased pressure of CO2, hemoglobin lowers its affinity for oxygen. And if we have an increased pressure of CO2, we have an increased affinity for oxygen. Let's take a look at a few of these in graph form. Here we can see the effects of pH on the shift of the saturation curve. Remember that the lower pH, in this case of 7.2, is going to lower oxygen affinity and have a shift to the right. The higher pH, that of 7.6, is going to raise the oxygen affinity and shift to the left. Here is an example of temperature. We can see in the middle line of 38 degrees Celsius, we have a nice, even, normal oxygen hemoglobin saturation curve. However, if we increase the temperature to 43 degrees Celsius, we are going to have a shift to the right, and therefore the actual oxygen affinity is going to be lowered. Conversely, if we decrease the temperature, making it cooler, 20 degrees Celsius in this case, we're going to shift the curve to the left and increase its oxygen affinity. The same can happen, the curve shift, for the pressure of CO2. Remember that an increase in the pressure of CO2 is going to shift the curve to the right, where a decrease in pressure of CO2 will shift the curve to the left. Carbon dioxide transport in the blood is a little bit more complicated than oxygen transport. Aerobic metabolism in the peripheral tissue is going to generate the waste product of carbon dioxide. After entering the bloodstream, carbon dioxide is transported in three ways. The first way, much like oxygen, is that it is dissolved in plasma. This is only about 7%. It has limited solubility in the plasma. The rest is going to be transported into the red blood cell, where it is going to be stored in two ways. 23% of this is actually going to be bound to the protein portion of the hemoglobin molecules, resulting in a compound that is called carbaminohemoglobin, or HbCO2. This is a reversible attaching to the exposed amino acid groups that allows it to be able to detach and remove itself from the body or attach when it's at the tissue level. The final is going to be converted to a molecule of carbonic acid where it undergoes this actual chemical process, which we'll get to. The process of actually storing and converting carbon dioxide to bicarbonate is as follows. We first need to take CO2 and combine it with water. This is a reversible reaction that is catalyzed by the enzyme carbonic anhydrase. Carbonic anhydrase makes this happen very, very rapidly. So we have CO2 plus water, and it gets converted into carbonic acid, which is H2CO3. Immediately upon this happening, the carbonic acid molecules will dissociate into the bicarbonate and hydrogen ions. So the H2CO3 will become an HCO3 plus a hydrogen ion. From there, the bicarbonate itself, the HCO3, is going to be exchanged out of the cell for a chloride ion in a process known as chloride shift. We'll get to why this happens in a bit. That hydrogen ion that's left over is going to stay inside the red blood cell and bind directly to the hemoglobin. The hemoglobin molecules function as pH buffers in the body. Here we can see a visual representation of the three different methods by which CO2, or carbon dioxide, is stored in the body. So CO2, remember, will diffuse into the bloodstream. From there, it's going to go 93% into the red blood cell and 7% will remain in the plasma. That 93% in the red blood cell, 23% is going to bind itself to the hemoglobin, again, forming carbaminohemoglobin. So this is actually going to be bound to the amino acid structure and not to the heme unit directly like oxygen is. The other 70% is converted to carbonic acid. Again, remember, this is CO2 plus water, H2O, that is catalyzed by the enzyme carbonic anhydrase to create H2CO3. That H2CO3 will then immediately dissociate into a hydrogen ion and the bicarbonate ion. From there, the hydrogen ion is going to bind to the hemoglobin, and the bicarbonate ion will leave the cell in exchange for chloride. This process is known as the chloride shift. Looking at the exchange of oxygen and CO2, we can see that there needs to be an equilibrium between them. The pressure of both of these gases are stable in alveoli and tissue. An example is that for every oxygen molecule entering the peripheral tissue, it is balanced by an oxygen molecule absorbed by the alveoli. Equilibrium is disrupted when the tissue oxygen demand increases. And to actually counter this, we increase respiratory rate and tidal volume. Without that increase, the alveolar blood and tissue oxygen levels will drop and the pressure of CO2 levels will increase. This leads to hypoxia and a drop in pH levels. This is actually a visual representation of the balance and exchange and storage of the different gases. First, we'll start with O2 pickup. Remember that oxygen is transported from the alveoli into the blood. Oxygen is going to have 2% of it that gets dissolved in the plasma, and the other 98% of it is going to reversibly bind to hemoglobin. It will then be transported. Upon being transported to the tissue, hemoglobin will then unbind itself from the oxygen. The oxygen is going to then move itself from the red blood cell, where it was bound to the hemoglobin, and from the plasma, and then go into the tissue. Carbon dioxide, we will actually start with pickup here. Pickup from the carbon dioxide is going to happen at the tissue level, where it will diffuse from the tissue into the blood. Remember that 7% of it is going to go directly into the plasma. The other 23% will actually bind itself to the hemoglobin molecule, and the final 70% is going to then convert itself into carbonic acid via the enzyme carbonic anhydrase, where it will then immediately dissociate into a bicarbonate ion and a hydrogen ion. Remember that the hydrogen ion will bind itself to the hemoglobin, and the bicarbonate ion will then exchange itself out for chloride via a process known as chloride shift. Now, I've mentioned this before. The bicarbonate ion being transported out of the red blood cell in exchange for chloride is facilitated via diffusion using an antiporter protein in the red blood cell. This transport process maintains electrical neutrality as a negative ion is exchanged for a negative ion. Both ions are moving down their respective concentration gradients. The bicarbonate in the blood can now serve as a buffer for pH regulation, and this is why the chloride shift is important. Once the red blood cell gets back to the lungs, the process simply goes in reverse. Here, the bicarbonate ion is going to go back into the red blood cell, chloride will go back out, then the bicarbonate ion will reattach itself to the hydrogen ion that was bound to the hemoglobin. From there, it will then convert itself back from carbonic acid back into CO2 and water. The CO2 that is bound to the hemoglobin directly will unattach itself, and the CO2 that was dissolved in the plasma will thereby leave the plasma and go back into the alveolar space. And then we exhale CO2. Now, even though the muscles driving ventilation are voluntary skeletal muscles, they are nevertheless driven by a rhythmic pattern of contraction and relaxation that is similar to the cardiac pacemaker rhythm. The difference is that because skeletal muscles must contract due to the action of motor neurons, the respiratory pacemaker must be located in the brain. The respiratory pacemaker is called the central pattern generator, or CPG. It is located in the medulla oblongata and the pons. The central pattern generator activity is modulated by sensory input that includes chemoreceptors, which are going to sense concentrations of CO2, oxygen, and hydrogen, and mechanoreceptors, which are stretch receptors in the lungs. Now, although it might seem surprising, there is still quite a bit we don't understand about the control of the ventilatory patterns by the brain. But here's what we do know. First, during quiet breathing or normal ventilation, for inhalation, we have activity that occurs in the dorsal respiratory group that stimulates the inspiratory muscles. This is going to cause the diaphragm and external intercostal muscles to contract and inhale. From there, the neurons in the dorsal respiratory group become inactive. They remain quiet for the next 3 seconds and allow the inspiratory muscles to relax. From here, of course, the diaphragm and external intercostal muscles relax and passive exhalation occurs. So this means the dorsal respiratory group turns on when it wants to actually breathe in, turns off when it wants to breathe out. Only the dorsal respiratory group is involved in quiet breathing. Forced breathing is a little different. Here we have increased activity in the dorsal respiratory group, which stimulates the neurons to inhale, and the ventral respiratory group. So the ventral respiratory group has inspiratory muscles. It's going to trigger the actual engagement of the accessory muscles that we talked about in the earlier lectures. So here, for forced breathing, dorsal respiratory group and ventral respiratory group will both turn on. The inspiratory muscles will then contract and expiratory muscles are relaxed, so inhalation occurs. Now, from here, the dorsal respiratory group will turn off and the inspiratory muscles that were activated by the ventral respiratory group will turn off. We then use the accessory muscles or expiratory center of the ventral respiratory group to actually activate the accessory muscles for exhalation or expiring. From here, after each inhalation, an active exhalation will now occur as the neurons of the expiratory center of the ventral respiratory group stimulate the appropriate accessory muscles. The inspiratory muscles will then relax, and the process keeps occurring. So if we understand that there are really 2 groups here, the dorsal respiratory group in the brain and the ventral respiratory group. The dorsal respiratory group only deals with inhalation or inspiration. It's going to modify its activity via chemoreceptors that detect O2, CO2, and pH levels in the blood and cerebrospinal fluid, and mechanoreceptors, remember, that deal with stretch. Now, it's mainly concerned with inspiration. That's all it deals with. So when it turns on, you're breathing in. When it turns off, you're exhaling. The ventral respiratory group has a main association with expiration, but it does have an inspiratory center. So it's going to have 2 different centers, one for breathing in and one for breathing out. When you're breathing in, you turn on both the dorsal respiratory group and ventral respiratory group, the inspiratory center for the ventral respiratory group. When you are breathing out, you then turn off the dorsal respiratory group, turn off the inspiratory portion of the ventral, and then turn on the expiratory portion of the ventral. So the ventral, remembers, in 2 parts. It has an inhalation and exhalation because the ventral respiratory group deals with the accessory muscles for breathing. Now, these centers are going to be located a lot in the pons that are going to adjust the rate and depth of respiration. So we can promote the inhalation by stimulating the dorsal respiratory group, and we can inhibit a lot of these other centers and promote passive or active exhalation with the pneumatic centers or the pneumotaxic centers. Now, there are higher centers of the brain located in the hypothalamus, limbic system, and cerebral cortex. Now, these can alter pneumotaxic centers to where normal breathing can occur without higher input. So we can actively control our breath, or we can kind of push it down to where it's controlled by a lot more simple systems. Now, one of the major reflexes that we need to discuss is the chemoreceptor reflex. Under normal conditions, this is very key. Under normal conditions, the pressure of CO2 is the most important factor influencing respiration. So how much CO2 that we actually have is going to determine our overall ventilatory rate, how much we breathe. So the respiratory centers do respond to a lower pH or a decrease in oxygen, but the decrease in oxygen has to actually drop below 60 millimeters of mercury before triggering a respiratory center response. This is simply a homeostatic flow chart that is going to show you how we can actually adjust the breathing rate based upon different sorts of disturbances. So at homeostasis, remember that our driving force is going to be arterial pressure of CO2. If we have an increase in CO2, a hypercapnia, we will thereby stimulate arterial chemoreceptors and we'll have an increase of pressure in CO2 in the cerebral spinal fluid as well as a decreased pH. This will cause a stimulation of respiratory muscles to actually increase overall activity, and we'll have a stimulation of chemoreceptors in the medulla oblongata. The goal behind this, we increase our respiratory rate, both our breathing rate and the overall depth and breadth of our tidal volume. This will cause us to get more oxygen into our body, eliminate more CO2, which is the key factor, eliminating more CO2, and thereby returning us to normal. Now, if we have a decreased pressure of CO2 or hypocapnia, here we have an inhibition of arterial chemoreceptors and a decrease in the pressure of CO2 in the cerebral spinal fluid. What do we do here? Simply the opposite. We inhibit respiratory muscles. We decrease the stimulation of the chemoreceptors, and it causes us to breathe less, thereby increasing our concentration of CO2 in the body and causing us to return to normal. Now, there are a few other protective reflexes that we need to look at, kind of weird breathing, so to speak, and that includes sneezing and coughing. Sneezing is triggered by irritation of the nasal cavity wall and is designed to expel something that is in the nasal cavity. Same case with coughing, and that it's going to be triggered by irritation of the larynx, trachea, or bronchi. And this is, again, trying to eliminate something that's in those passageways. In both cases, we are involved in a period of apnea. Now, this is a period in which no breathing has stopped, and it's followed by a forceful exhalation. So it's just designed to clear irritants. Since I usually get asked about it, hiccups are going to be an irregularity of breathing to where the body's trying to reset itself, and it can happen typically by upsetting some of the normal rhythmicity of breathing. Finally, we need to break the system. And here we're actually looking at pulmonary diseases. The major pulmonary disease that we look at in the overarching theme is chronic obstructive pulmonary disease, or COPD. This is a general term for a progressive disorder of the airway that restricts airflow. Now, the major symptoms of COPD are shortness of breath, irregular breathing, and a chronic cough, usually with a lot of sputum production. This is like saliva, mucus, and things like that. There are three specific examples of COPD. The first is asthmatic bronchitis, or simply asthma. This is a term used when symptoms are acute and intermittent. The condition is characterized by conducting passageways becoming extremely sensitive to irritation to where they can become inflamed, thereby constricting the airway and increasing mucus production. Typically, this is something that you're born with and you grow out of, but it can also be triggered by different allergens, toxins, and in some cases, exercise or increased stress. Asthma is typically dealt with by using different corticosteroids to reduce the inflammation in the form of an inhaler. Another form of COPD is chronic bronchitis. Now, chronic is the key word there, as it means it's the long-term inflammation and swelling of the bronchial lining leading to an overproduction of mucus. This can clog airways, thereby increasing the resistance, so decreasing flow. The characteristic sign is frequent coughing with increased sputum production. Typically, the cause of this is from cigarette smoking. There's also environmental causes. If you grew up in a more air-polluted city, that can typically cause it as well. This often leads to chronic bacterial infection of the lungs that needs to be dealt with. Here's an old man coughing, symboling chronic bronchitis. And finally, we have emphysema. Now, emphysema has been discussed before. It is a chronic progressive condition where we have an increased overall compliance but a decreased elasticity. The alveoli gradually expand and merge with adjacent alveoli, and it forms large air spaces but with little elastic connective tissue to allow for recoil. So because of this, we have a loss of respiratory surface, and that decrease in surface area restricts oxygen absorption. So you have chronic shortness of breath and the inability to actually exhale very forcefully.